Mm. My name is Neil, and I want to take you down a journey. I want to tell you a story. It's a story that begins with some minerals that look a little bit like those pictured on the screen and goes through all the way through to the research that I'm doing today. And that story takes about 4 billion years, although I do aim to try and tell it a little bit quicker. But not only does the story cover a vast amount of time, we also cover a vast amount of distance. We cover the distance during the story as a journey from the sun all the way through to a process that happens within ourselves. And that's a journey that covers about 149 million kilometers. So let's start off by going through this journey of time. Our journey starts off about 4 billion years ago. The planet Earth is covered in sea. There's very little land, of any land mass at all. And underneath that landmass, we start to see the formation of outcrops of a mineral called serpentine. I actually have some of the serpentine here. I have some in my hand. You can't, oh, try to put it in the camera. You can't feel it, but it's a very soapy mineral. And this mineral is formed when the alkali gases coming up from the core of the Earth start to interact with a rather acidic environment. And 4 billion years ago, our sea was very, very acidic. So we had this formation forming, as I say, from these alkali type materials coming up from the Earth's core and then coming through the Earth's crust and interacting with this very acidic ocean. And across the surface of this mineral, something starts to happen. We start to see something happening with protons and electrons. A theory that was first put forward as to what energizes human life by a chap named Peter Mitchell, and I'm not talking about the Pete Mitchell from the Top Gun movies, that would be Maverick, but Professor Peter Mitchell, who in 1961 first proposed what he called the chemiosmotic theory, and he then published that theory in 1966, and in 1978, when his theory was proven and shown to be correct, he was awarded a Nobel Prize. What the chemiosmotic theory basically says is that protons inside of us across our mitochondria, we'll talk more about this later, are pumped across a membrane, and that pumping across the membrane creates the energetic forces required to drive electrons down a chain, down the surface of the membrane, and also to power a fantastic little turbine, a little machine that sits inside all our mitochondria, called the ATP synthase, or complex 5 of the ETC, and we'll talk more about that a little bit later. But fairly recently, in 2023, a team of scientists actually showed that this proton pumping process occurred long before anything organic existed on our planet. It occurred across this mineral. It occurred across the serpentine mineral, um, where these outcrops were formed, what they call these white smokers. So not the black smokers that were originally thought to be the origins of life, but the white smokers which exist in far more shallow waters. And there's actually one not too far away from where I am in England, because there's one, I think it's called the Lost City, which sits just off the coast of Ireland, and where we've been able to test out some of these theories. Is quite important for me to point out as I tell the story that I'm presenting here some evolutionary theories, and evolutionary theories will always remain theories, because the only way to prove that they're actually 100% correct would be to contact Doc Martin and Marty McFly and get ourselves a DeLorean kitted out with a flux capacitor to be able to go back in time and actually look at that. But according to this theory, and it's one that seems to be pretty validated, this proton pumping happens across this mineral. It happens as we can see. So what we can see in the frozen video screen here is the hydrogen ions in red going in and out of the membrane, going across from the alkaline inside of the mineral, and then coming back out with the acidic ocean. And while that's happening, it's actually driving the blue particles, the electrons, along the surface. And while this is happening over a period of billions of years, as I say, this is inorganic, a layer of fat starts to form on this mineral, hence the the soapy nature of the texture of serpentine. A soapy layer starts to form, And with the time, with evolutionary pressure, what actually happens is some of this lipid layer forms a bubble, and it breaks away from the rock. And what that means is that this proton pumping mechanism continues to operate, but now it's no longer going in and out of this mineral rock. It's now going in and out of this lipid membrane. And this lipid membrane is now free to float in the ocean. It's free to try to find a better source of these electrons closer to the surface where there's interaction from photons from the sun. And that is the birth of organic matter. Those little bubbles become the early bacteria and archaea and single-celled organisms on Earth. So this chemiosmotic process started in this inorganic manner and then evolved to the point where it could peel away from the rock and continue free floating in the ocean. So we can see that happening in this particular video over here, which is a very simple video. And what we see happening over here is two of these single-celled organisms floating in the sea. One depicted is a bacteria, and the other is an archaea, an ancient form of bacteria. And what happens over time, what the evolutionary theory proposes, is that at some point a bacteria hitched a ride on an archaea. They formed a relationship whereby one was attached to the other. And what happened was the archaea then gave up its ability to do this chemiosmotic process and thought, I'm going to leave the bacteria that's attached to me to do that, and I will then harvest the energy that is being generated by that bacteria, and I will use that to power my own proteins and my own relation and my own processes, the processes that ultimately become DNA, the proteins that become DNA. And this relationship then becomes fully symbiotic. At some point, as we go through here, what happens is that the archaea actually envelops the bacteria. So now the bacteria actually exists inside the archaea. But that proton pumping mechanism, that chemiosmotic process, now continues to operate within or by the bacteria, or what was the bacteria. And this is now happening across an inner membrane, a membrane that is inside the archaea. This then becomes the basis of a eukaryotic cell. So eukaryotic cells, mammalian cells, etc., use these karyotic cells, but so do fungi, so do yeasts, contain these eukaryotic cells. And this process of pumping hydrogen ions, of pumping these protons backwards and forwards across the membrane, is so evolutionarily perfect that it doesn't really get upgraded. From the time this first happened, so originally inorganically 4.5 billion years ago, to the point where it evolves inside a eukaryotic cell, or I think, remembering offhand, that's about 250,000 years ago, the process doesn't change. So we see this as a common process, as I say, from anything from yeast to the mitochondria within us. And that kind of leads on to where this goes, because what happens with this process is it evolves to take place within our mitochondria, within the mitochondria of eukaryotic cells, and we are made up of eukaryotic cells, as I say, in the same way that certain fungi are, yeasts are, etc., made up of that. And these mitochondria, which use this chemiosmotic process, sit inside our cells. So we have multiples of them. Different cells have different amounts, from a couple of hundred in certain cell types to a couple of 100,000 in other cell types. But all of our cells contain mitochondria, and we are made up of trillions of cells. But here comes something really, really interesting. We have now come to realize that these mitochondria talk. They talk to each other. So all the mitochondria within a cell are constantly communicating through chemical and electrical signaling with each other. But more than that, they're not just communicating with each other inside the same cell. They're communicating with each other across our cells. So the mitochondria in our gut are sending signals that are being read by the mitochondria in our mind, and the mitochondria in our mind are sending signals that are being read by the mitochondria, or being understood by the mitochondria within our muscles, etc. And more, we're also coming to realize that through the interactions of how we behave, not only are our mitochondria potentially communicating with each other within ourselves, but our mitochondria could well be communicating with other mitochondria in other organisms. Not directly, there's no language of mitochondria, but our mitochondria are generating the chemical and electrical impulses that cause us to behave and respond and see and do things in a certain way. And that is then perceived by the senses and of another individual, which in turn becomes electronic and chemical signals within that individual, which go back into their mitochondria. So we have this idea that mitochondria are communicating with each other throughout us, but also potentially across living organisms or across living creatures. So that story really covers this 4.5 billion years of this chemiosmotic process that's taking place across an inorganic surface of serpentine, of this mineral, 4.5 billion years ago, to being a process that is so perfect evolutionary that it has hardly changed in time and still takes place within the mitochondria inside all of us. So that really looks at the story over time. But let's look at it over distance. All right. So what am I talking about when I talk about distance? Well, those electrons that are passing down our electron transport chain, they originate in the sun. They originate in the sun, and they become photons that escape from the sun, or they become part of the photons that escape from the sun, traverse space in all directions, and some of those cover the distance and strike Earth. And here on Earth, something really happens. Interesting happens in that within our plants, within our green leaves, wherever we have the ability to harness sunlight for energy, so photosynthesis, those electrons are harvested, and they then are removed and they become free, and they become a little bit unhappy about that, and I'll cover that in a moment. They're not happy once they're removed, but we take those electrons, the photosynthesis within the electrons turns some of those electrons into glucose, the molecule shown on the screen over here. We consume some of those glucose directly when we eat fruit and vegetables, but others of those glucose get consumed by animals, pictured as a cow, but all wild stuff. The cows then sometimes store some of that in the form of glucose. Others get converted to fat and also then gets used to drive certain other processes. Ultimately, we consume those glucose in the form of glucose or in the form of other energy materials, but particularly glucose or fats. And our body then goes through a series of chemical processes, glycolysis within our cells, whereby we break that glucose down, or we break the food down ultimately to the glucose molecule itself. And then the glucose molecule goes through the process where it gets the electrons from that glucose, gets broken back out of it. Those electrons that originated out of the sun get broken out and they get attached to something called NAD. So this can happen within the cell's cytosol using glycolysis, but more particularly we see this happening within the Krebs cycle of our mitochondria. So I'm not going to go through the details of the Krebs cycle. I'm not going to take you back to school, but you can see there are multiple phases within the Krebs cycle where you see this NAD being reduced. In other words, electrons are added to NAD. It's what reduced means, to form NADH. So this cycle here, NAD to NADH, and that's the reduction. You can see in the Krebs cycle it happens here, it happens here, it also happens here. Also not pictured, but over here we see a similar process happening, but where we actually see FAD, so the electrons being attached to FAD, or FAD being reduced to form FADH2+. And then that process, those chemicals, those carriers, that NAD and NADH, which is now reduced because they've now got those additional electrons, those get passed to the electron transport chain within our mitochondria. And at that point, they get oxidized. So basically the electrons, along with the hydrogen ions attached to the NAD and the NADH and FADH, gets broken off. The hydrogen ions, the positively charged protons, get pumped across through the complexes to the other side of the inner mitochondrial membrane, forming an energy difference. And this is Pete Mitchell's chemiosmotic theory. This is what we're seeing here, this process that started 4 billion years ago inorganically, taking place within our mitochondria. But these electrons now get put into the electron transport chain where they get passed using cytochrome B and C, etc., down the electron transport chain. And remember I said earlier that from the time these electrons get photosynthesized in green leaves or the grass, they become a little bit unhappy, they're a little bit miserable. They feel like they need to complete their journey. Well, guess what? Inside our mitochondria, inside complex 4, those electrons finally find peace, because those electrons get combined with some of the oxygen that we're inhaling, so part of our respiratory process, and some of the hydrogen ions that are still here, and they form water. And that is where the electrons find peace. In water, when those electrons are brought back and combined into the oxygen and the hydrogen to form water, to form a water molecule, they have found peace. They have reached their final destination with perspective to our story. In terms of the chemiosmotic process, this charge difference between all these positively charged hydrogens on the outside of the inner mitochondrial membrane and the negatively slightly charged environment on the other side causes balance. We want to see balance, and through complex 5, through this fantastic little turbine motor that exists inside our mitochondria, those hydrogen ions get pushed through to find balance. The positive charge gets pushed through to find balance in the negative. That spins round a little turbine, and that turbine attaches an inorganic phosphor molecule to a compound called ADP, adenosine diphosphide, diphosphide meaning 2 phosphors, and by attaching the third one, we create ATP, or adenosine triphosphate. And ATP is the currency. We talk about it being the energy currency of the cell. Effectively, that is the chemical form of energy that our cells can use for life. That is what powers us. So we have these two stories. We have this distance of time as the evolution of the chemiosmotic process over 4 billion years. And we have the story of distance, these electrons that start their life inside our sun and then traverse space, being unsettled as they get photosynthesized as part of the photosynthetic process within plants, only to arrive inside the mitochondria of a eukaryotic cell in a living organism such as us, and to be reunited with all the other bits and pieces to form water and create happiness for them, a nice stable environment that I am intentionally anthropomorphizing. And of course, in this process, we generate the ATP required for life. But we don't just generate ATP. We also create a whole bunch of other metabolites, things that affect how our DNA is methylated, and histone acetylation takes place. In other words, what our gene expression is. And we form all sorts of other little bits and materials that are used for this chemical interaction that allows our mitochondria to dictate the health of our bodies and dictate the way we respond to our environment. So that scientific story then brings me to what I'm specifically looking at in my PhD. What part of this journey over time and space am I specifically looking at? So I'm looking at that glucose molecule and how the chronic exposure to that glucose that we experience on the Western diet, pictured in some of the foods over here, but in lots of other foods. And please be aware, I'm referring here to chronic exposure that's in our diet, not acute exposure. We do require some glucose in our bodies, but it's the chronic exposure of all the glucose that we're continuously consuming in our current lifestyle. And I'm looking at how that chronic exposure affects our mitochondria such that it results in the chronic diseases that we see today. Why? What is the relationship between all this glucose and our diet and the fact that our health span has decreased so low that in England the average health span is now 62 years. If you consider that average lifespan is 85 years, predominantly being held there by the fantastic advances in modern medicines, but our health span is around 61 to 62 years. That means that we are living, so the definition of health span is the number of years that we live disability free. So if we stop living on average disability free at age 61 to 62, but we're going to live to 85 years, that means that a good potential of our life, we are going to operate being supported by drugs, potentially, and all those drugs have harmful side effects. There isn't any drug or medicine that we take today that is actually designed to be taken for a period of 20 years. All right. So all drugs have some sort of side effects, obviously to a greater and lesser extent. And ultimately, this chronic glucose exposure potentially leads to a premature visit from this guy. And that's not what any of us want. So that is what my research is specifically looking at. It's the relationship between the chronic glucose in our diet and the reduction in health span and the exposure that creates to make us vulnerable to chronic diseases and ultimately to death. It's really worth knowing that I'm not the only one. My research is just one part of a much bigger story, and that bigger story is being pieced together by different labs all over the world. This is such an interesting picture on the left-hand side. This is looking at the number of publications across different organelles over a period of time. And the line that we're interested in is this brown line over here. And we can see that by 2020, and I would love to see where this sits now in 2025, because I suspect it would have gone up even higher. We can see the number of publications, the number of research that's being conducted in understanding mitochondrial bioenergetics. And we can see if we follow this dotted line here, this is kind of the research looking at the nucleus, which is kind of looking at DNA. And you can see that research really peaked in 2001 when the Human Genome Project completed the mapping of the human genome. And the science community thought that understanding that would allow us to revolutionize health. We thought we could just detect which genes were responsible for certain chronic diseases and remove them or do something to downplay them. Or take them out using gene editing, etc. But you can see that actually the amount of research purely looking at this has actually been on the decline. It's still very high, don't get me wrong, and it's still very important. But you can see it's been on the decline. And the main reason for that is that, try as we might, we've discovered that there's just no one gene responsible for one thing. We know if we block certain genes responsible for cancers, the cancers happen anyway. So there's a lot of redundancy within our genetic material, genetic code, and there's a lot more than just one gene responsible for chronic diseases. There's a multitude of them. Right? So what we have realized is that our genetic makeup is not necessarily on its own the part of the puzzle that we need to tackle. We need to look at not just which genes we have, but which genes are expressed. Which are the genes, the epigenetics, which are the genes. So just because I carry certain genes, let me try and put this in a simple perspective. I might carry all the genes that predispose me to lung cancer. But if I don't smoke, and I don't engage in the other activities that make that gene become expressed, that epigenetic expression of that gene, I probably won't die of lung cancer. However, if I am a smoker, which I'm not, or if I engage in other unhealthy activities, I might then bring that gene expression up. And again, we've now been able to show that it's the mitochondria that fundamentally do the programming of our methylation cycles and our histone In other words, it's our mitochondria that translate all the signals coming into our bodies, and based on that signals, the mitochondria respond by dictating which of the genes that we do have are the ones that become expressed. And as I say, I'm specifically looking at the role of glucose in this, or chronic glucose exposure, but this is a big field of study. It's a wide field of study. So here comes the important part. What part can you play in helping with this story, with this research, with the kind of research that's going to make you healthier, make you live longer? Because ultimately the chronic diseases that are affected, we all know. I don't think anybody watching this video has not had a direct relationship, either themselves or through a friend or loved one, who has not has not dealt with cancer, dementia, heart disease, obesity, diabetes. The list goes on. And this research and understanding our mitochondria affects all of those. However, there are powerful forces that affect where the funding for this research goes. And two of those very powerful forces, ones that influence government and influence funding bodies dramatically because of their clout, are the food industry and the big pharma and the pharmaceutical industry. And as you can imagine, the kind of research that I'm doing, the kind of research that says maybe if we change the formulation of the food that we eat, or we educate people properly around the food that we eat, we can then reduce the disease. Just think about the impacts of that. Just remember, okay, and I want to make sure that I'm not really touching too much on, I know that those within certain communities would say, well, the big food company and the big pharmaceutical companies are working in cahoots. My personal belief is that these two things are driven by the same thing. They're driven by profit. Big companies, or all companies, big companies are not answerable to their customer base. They're not even answerable to their staff. Companies are legally only responsible to their shareholders, and shareholders only want to see return. They only want to see profit. That's why they invest. So these two companies, I think, have evolved. These two industries have evolved to support the ability to maximize profit. And in so doing, they've formed a fantastic symbiotic relationship. But I don't think that symbiotic relationship was formed by people from both companies sitting behind a desk or in a meeting room and saying, this is what we're going to do. We're going to plan this. I think it's just evolved that way. All right. But research like mine affects both those big industries. So if I could show, and myself not only, but my research along with the big puzzle that I spoke, we can show that we need to reformulate food to reduce the impact of chronic glucose exposure. That's going to reduce the bottom line of the food companies for some time because they're going to have to reformulate and they're going to have to change certain things. And at the same time, if that happens, and it does happen, and we start to make people healthier, well, that then in turn is going to affect the profits of the pharmaceutical companies because people are going to get less sick. So we're going to see less chronic diseases. There'll still be acute diseases, but we'll see less chronic diseases. And what that means is that the kind of research that I'm doing, the kind of research that will positively impact you and the people you know and the people you love, battles to get funding. It's not impossible, but it's really difficult. And that's why this funding page has been set up. This is the part that you can play. It doesn't matter if you sponsor my research with a $5. It doesn't matter if you put in 10 quid, 50 quid, 100 quid. And I'd love to see some corporates who want to see the benefits and health impact through their social impact funding. I'd like to see those corporates put in bigger amounts because improved health span and improved health improves productivity. And improved productivity actually helps the bottom line of all the other companies that potentially aren't the food industry and aren't the pharmaceutical industry. In other words, your small business, your medium-sized business, your large enterprise. Your staff affect your bottom line by their ability to deliver energy. It's all about energy. That energy that starts in the sun and comes through into our mitochondria is the same energy that drives your productivity, that drives the way your business operates. And by contributing some amount to my research, you contribute towards your productivity, but you contribute to the health and well-being of your staff. And as individuals, every $5, every 10 that you contribute to my funding is a positive impact on your story, because this is your story. It's not just my story. It's your story. It's all of the stories. So I just want to say thank you for looking at this page, for watching this video, for considering donating or just sharing. If you want to contact me directly because you want to find out more, you're welcome to email me at the email address that's on the screen. And you can also catch up with me on LinkedIn. You can direct message me if you wish, or just look at my posts. And I'm really happy to answer any questions that you put forward that maybe will help you to come to a decision to contribute towards my funding, to contribute towards this research. And I really, really look forward to hearing those questions and interacting with you. And I thank anybody that's going to donate in advance for your contribution. It is important and it is greatly appreciated. Thank you for your time.